Okay, today in Honors Biology, we're going to start Chapter 8, which deals with DNA. So we're going to talk about DNA replication and then protein synthesis. So first off, let's refresh our memories on what nucleic acids are. So nucleic acids include DNA and RNA, so we're going to talk about both of those in this chapter. Both are polymers made up of repeating monomers called nucleotides. So remember, a polymer is a large molecule, has many repeating units called monomers, and in this case the monomer would be a nucleotide. DNA carries the code for the genetic makeup of an organism, plus it carries the code for many of the proteins it will make. It is also involved in making RNA, which is then used to build proteins. The reason proteins are very necessary because they make up enzymes that control all the chemical reactions in a cell, in addition to be used for other things. So let's talk a little bit about DNA in detail. So DNA actually stands for deoxyribonucleic acid. Its structure, it is an organic molecule, meaning that it's going to contain carbons. It is double-stranded, made up of nucleotides, and forms a helix, and it contains units called triplets. So triplets are 3 bases. The nucleotide of DNA specifically is composed of a pentose or 5-carbon sugar called deoxyribose. So remember, if it ends in "-ose", it means it's a sugar. There are sugars that do not end in "-ose", though. It also contains a phosphate group, or a PO43-, and then a nitrogen-containing base. The nitrogen bases occur in 2 categories. We have purines and pyrimidines. Your book should show you examples of both. So purines are double carbon rings, and they include the nitrogen bases adenine and guanine. So when we draw DNA, I'm not going to write out the word adenine, guanine. I'll use an A for adenine and a G for guanine. The pyrimidines are single carbon rings, and they include thymine and cytosine. So when we do DNA, we'll put T for thymine and C for cytosine. A gentleman by the name of Erwin Chargaff determined that all organisms contain these bases, but they appear in varying amounts, and further determined that the amounts of adenine will always equal the amounts of thymine, and the amounts of cytosine will always equal the amount of guanines, and so that's called Chargaff's rule. Now, the double helix form was discovered in 1953 by Watson and Crick. I think Watson was maybe an American and Crick was from England. I may have them backwards. They also used X-ray, the X-ray crystallography work of Rosalind Franklin, along with Maurice Wilkins. And there's a picture of the X-ray crystallography on page 222 of your text. The phosphates and sugars are covalently bonded to each other, and they will form the sides of the molecule. Then coming off of the sugars are your nitrogen bases, which are hydrogen bonded to the opposite nitrogen base and form the rungs of the ladder. DNA molecules have very distinct ends. They have a 5' end and a 3' end. Okay, so replication of DNA. So if you'll remember, back in mitosis and meiosis, we said your DNA had to replicate in the S phase, right? So replication is carried on by enzymes and other proteins. The 2 strands of DNA are complementary to each other, so that simply means if you know one strand, you can predict what the other strand is. And they'll ask things like that on the EOC. The base sequence on one strand will determine the sequence on the other strand. Enzymes are needed. They will break the bonds that hold the 2 strands together and will replicate both strands of the molecule, and replication will always occur in the 5' to 3' direction. Now, here are the names of some of the enzymes. So keep in mind, if it ends in ACE, it's an enzyme, although not all enzymes will end in ACE. So DNA helicase breaks the hydrogen bonds on the bases, or between the bases, and basically unzips the DNA. Then DNA polymerase will bring in free floating nucleotides and attach them to each other at the 3' end, so that the new strand of DNA elongates only in the 5' to 3' direction. Along one strand, a new strand is made in one continuous piece towards the Y junction. This new strand is called the leading strand. To make the other strand, DNA polymerase must work along the other template strand in the other direction, going away from the Y-shaped junction of the strand. This new strand of DNA is called the lagging strand and is made up of structures called Okazaki fragments, which are then pieced together by an enzyme called DNA ligase. And then, in addition, DNA polymerase will also proofread the molecule as it replicates. Now, DNA molecules typically contain hundreds of origins of replication points, or ORIs. This allows for quicker replication of all the genetic material. DNA replication is also said to be semi-conservative, which simply means each DNA molecule contains one of the original strands and one of the new strands. Now, I thought about drawing replication out, but I will draw it on the board, and then I will have someone send you a picture of it. Here's a DNA molecule, and it's just going to show you the leading and lagging strand, but it doesn't show you the basis. So it's important that when you get that picture sent to you that you draw it into your notes. Okay, so here's your DNA helix. You can see that you have a 3' end and then a 5' end, okay? So when you're making a new DNA section, you always start on the 3' end of one of the original molecules so that you can build the new DNA molecule from the 5 to 3'. This piece here is being made continuously towards the Y junction, and so we would call this the leading strand. Over on this side, so you're replicating this side and you're also replicating this side. On this side, so this is your 3' end, so you're going to build it because you want to build it in the 5 to 3, so you're going to start towards the Y junction and make your DNA in this direction. And then it's made in little pieces called Okazaki fragments. And then here it shows you kind of a picture. Here's an Okazaki fragment. Here's one. And then those fragments will be joined by DNA ligase. Okay, so once again, to save time, I'm going to have someone send a picture to you of DNA replication that we draw in class. Okay, so that takes care of this section, so that will go with your first study guide on DNA and DNA replication.